Hey everybody, what's going on? How are we? Let's get after cell signaling today and talk about how cells communicate. A lot of times we think of cells as sort of individual things because they have the cell membrane that keeps things in and keeps things out, but the cells actually do need to communicate with each other and they need to be able to send messages between one another. So that's sort of what we're going to talk about today, is how cells send a message and mainly how they receive the message and interpret that information. Anytime a cell is talking to another one and a signal is sent from cell A to cell B, that's called a signal transduction pathway. And the way that a cell sort of interprets a signal and uses the signal is going to determine how well that cell can maintain homeostasis. If certain parts of the cell don't accept the signal the right way, then the cell won't be doing the things that it needs to, because those signals are sort of necessary for the cell to carry out the processes that they need to maintain normal functioning. So first, let's talk about the different types of signals. And different types of signals just has to do with where the signals are coming from and going to. An autocrine signal means that a cell is talking to itself. And auto means self, so it's a self signal. It's a signal that a lot of times tumor cells will release those to themselves. It tells them, hey, divide right now. And the more the tumor cell divides, more new daughter cells there are. And if each one of those is sending themselves more signals to divide, that tumor is just going to get bigger and bigger. Paracrine signals, well, para means nearby. Paracrine signals happen when cells talk to cells that are nearby each other. And neurotransmitters are kind of like the classic example of this, because neuron A talks to neuron B by sending out chemicals out of the cell, and it happens through a process called exocytosis, which we talked about in the last set of slides on diffusion. Hormones are cells that originate in one tissue and go off to a really far cell from the origin site. Your pituitary gland sits up in your brain, but it sends growth hormone out to all the cells in your body. So these three different types of signals allow the cells to communicate and tell them what processes to do. Now, with everything in your body, we have to regulate things. And a lot of times, this regulation is caused by something called allosteric regulation. And if you remember when we talked about enzymes a long time ago, when we were talking about our macromolecules, enzymes can be allosterically inhibited. Allosteric regulation happens when a molecule binds to a protein and the protein changes shape. And as that protein changes shape, it's going to change what the cell actually does. It may open a channel, it may turn something off. It just depends. But allosteric has to do with that cell changing its shape. Signals can be short-term, they can be long-term, it just depends. If the signal is near the receptor and it can stimulate it, it's going to keep stimulating it until that signal gets pulled away by something else. So it just sort of depends on what the process is going to determine how long that signal is going to stay in the receptor. Now, when we talk about cell signaling, three things always have to happen. No matter what kind of signaling we're talking about, these three things are going to go on. First thing that's going to happen is your ligand is going to attach to your protein. And if you remember, ligand is code word for signal. That's just the bioterm that we use. So this ligand right here is going to attach to the protein. As soon as it does it, and by the way, it attaches in the receptor is a point on that membrane protein that's going to receive the message. Once it does that, the protein changes shape. Well, anytime you bind a molecule to protein, depending on the charge of the molecule and whatnot, it's going to cause changes in the R groups, and that protein is going to have some kind of reaction to it changing. Once that takes place, that change of shape is going to cause some kind of response to happen on the other side of the protein, or inside the cell. Now, that can be reversed. A process can cause a change in the shape of the protein and so forth. The receptor can let go of the ligand and everything will stop. But these three things always have to happen. Signal attaches, protein changes shape, and there's some kind of response within the cell. So we sort of talked about this earlier. If the ligand is always on the receptor, that protein stays in the change shape and it keeps stimulating inside the cell. So that means that the signal is going to keep going and going and going. But let's say there's something blocking the receptor. Let's say something's already sitting in there. Can the ligand get in? No. And if the ligand can't get in, then the signal stops right there. It never even takes place. And when something's stopping the signal from taking place, we always call that an inhibitor or an antagonist, something that's going to stop that reaction from happening. When I look at these reactions, I sort of think of them as like dominoes. Once you hit that first domino over, everything else goes over. But the last domino can't go before all the ones in the line up to it get knocked down by each other. So you have to go from step A to step B to step C. And that's sort of how this signaling thing kind of works in here. When we talk about receptors, there's a couple different receptors. Cytoplasmic receptors, we're going to find those inside the cell, in the cytoplasm region. So the signal is usually going to be coming from the extracellular environment, and it's going to, if you have a cytoplasmic receptor, it's going to have to get through that cell membrane. Well, remember, that cell membrane has that lipid bilayer that's not going to allow polar things to go right past it. So the ligand for a cytoplasmic receptor is going to be small and nonpolar, because those things are going to have to be able to get through the cell membrane. Estrogen is kind of a classic example of something that uses a cytoplasmic receptor, with estrogen being a hormone released and the receptor being inside the cell that's going to be stimulated. Membrane proteins are usually going to be transmembrane proteins, so you'll have an extracellular domain and an intracellular domain. That membrane receptor is going to come in three different forms. One of them is an ion channel, and an ion channel is exactly that. Ions can get to flow through the channel, and when the ligand attaches to the ion channel, it opens up that protein, and things like sodium and potassium can cruise through. And if you look at how a neuron works, sodium-potassium pump is super important in moving those ions around. We'll talk a little bit more in depth on protein kinase receptors and G-linked receptors. They're not super complex, but they're not super easy either. So you just sort of got to pay attention and think about what they do in order to understand how they work. With the protein kinase receptor, it requires ATP. So the ligand, in this case, it's going to be insulin, is going to attach to the membrane protein right in that receptor. Once that happens, ligand attaches to protein, protein changes shape. As that takes place, ATP gets converted to ADP. Hopefully you know that ATP is adenosine triphosphate and ADP is adenosine diphosphate. The difference between tri and di is 3 phosphates compared to 2 phosphates. So if that frees up a phosphate, that phosphate can stimulate something within the cell. That thing that's going to stimulate inside the cell is called the target protein. So the insulin signal goes into the receptor that causes a change in shape of the membrane protein. ATP in this case has to break down, and the phosphate that comes off the ATP is going to stimulate the target protein. Once that target protein is stimulated inside the cytoplasm, that creates the cellular response. Remember, all three of our things happen. The signal goes in, the protein changes shape, there's a response within the cell. That's always going to happen in all of these scenarios. The next thing we'll talk about is a G-linked protein, and G-linked proteins are slightly different. In a G-linked protein, there's three parts of the G-linked protein. One, you have your receptor on the membrane protein. Next to that receptor and that protein that's in the membrane, you have something called a G protein that has GDP and GTP on it. GDP is similar to ADP, and GTP is similar to ATP. So these are high energy molecules just with a different base. Instead of adenine, you have guanine as the base in those ones. And then we have the effector protein, which is farther down the membrane. The G protein is actually going to move from the transmembrane protein that takes a signal, and it's going to move all the way over to the effector protein and cause something called signal amplification. If you look at the word signal amplification, amplification means to make something really big really fast. And that's exactly what happens with these G-linked proteins, is that one small signal becomes really big pretty quickly. And we'll sort of see how that works. So here's our basic synopsis of the beginning of the G-linked protein cycle. Again, where do we always start? Ligand attaches to the receptor. That's the first thing that has to happen. And we look at the shape of the receptor right here. As soon as that ligand attaches to it, notice what happened to that membrane receptor. It grew this little extra tail, and now it's touching this G protein. These three green things are going to be your G protein. When we look at the G protein in the first picture, we see GDP on there. Once that G protein gets stimulated by the membrane protein, GDP is traded out with GTP. GTP is stored energy, and that stored energy is going to allow this G protein to move down the membrane and get to the effector protein. We'll talk about a specific kind of effector protein when we talk about the cAMP pathway, but that's going to create turn a reactant into a product and cause that signal amplification. Amplification means that signal is going to get exponentially. It's going to exponentially increase the response within the cell. When the signal is amplified, another term for that we can use is called signaling cascades. Anytime you start with one small signal and it gets bigger and bigger and bigger, that is going to be called a signal cascade. The signal cascade requires something called a secondary messenger, or second messenger. A second messenger is a molecule that gets stimulated by that cascade within the cytoplasm. The example that we're going to look is called cyclic AMP, or C-AMP, or C-cyclic AMP, at adenosine monophosphate. So we've talked about adenosine triphosphate today, adenosine diphosphate, and now this one is adenosine monophosphate. And we're going to see how this reaction happens right here. The first thing that's going to happen, and we're going to use epinephrine as our example. Epinephrine is science word for adrenaline. And if you've ever been in a fight or flight situation where you either have to stand up and face something that's kind of scary or run and try to protect yourself, that puts you in a fight or flight response. Fight or flight, the big thing that you need is your body wants available energy, because this is kind of an innate prehistoric almost reaction inside of us that allows us to sort of protect ourselves. So the first thing that's going to happen, the G protein gets remember the epinephrine is going to act as our ligand. It attaches to the receptor protein. The receptor protein changes shape. It stimulates a G protein. The G protein moves over to something called adenylyl cyclase. Adenylyl cyclase is going to be the effector protein. That's the one that's just a little bit farther down the cell membrane from that membrane protein that was stimulated by the epinephrine. Adenylyl cyclase is going to make C-AMP, and it's going to make C-AMP out of ATP. Well, if ATP is adenosine triphosphate and C-AMP is adenosine monophosphate, that's going to leave a couple extra phosphates out there that can phosphorylate some things as we move down. Once C-AMP is created, it's going to bind to and activate something called the enzyme protein kinase A, and that's going to be inside the cytoplasm. And this is where that signaling cascade is going to start. Once protein kinase A is activated, it's going to phosphorylate two things. The first thing it's going to phosphorylate is something called glycogen synthase. Hopefully, you see that name and you recognize ASE. That tells you it's an enzyme. Well, synth tells you stuff's going to get put together, and glycogen is stored carbohydrate. So glycogen synthase, the enzyme that puts together glycogen or stored carbohydrate, look at what happens when it becomes phosphorylated. It gets inhibited. We're going to turn off the glucose storage mechanism. So the result, glucose isn't being turned into glycogen. Glucose isn't being stored. That's going to leave more free glucose inside our cell that can be transported into our blood. The other thing that gets phosphorylated is phosphorylase kinase. That one's actually going to become activated. And the cool thing about that is phosphorylase kinase is going to liberate glucose molecules from pre-made glycogen. So you have glycogen stores within your cells. The phosphorylase kinase is going to take those glycogen stores and break them down for you and turn them into individual molecules of glucose. Well, let's think back to what glucose is. OSE is a carbohydrate. Carbohydrates are your body's preferred source of energy. So if you're in a fight or flight situation where you might have to protect yourself, you're going to want that extra free energy sort of floating around your cell and into your blood. And remember, we started with one epinephrine signal. Well, look at this picture right here. Look how many blood glucoses come out of that. One epinephrine signal hooks to the receptor. The receptor changes shape, stimulates a G protein. G protein stimulates adenylyl cyclase, your effector. Your effector turns ATP into C-AMP, and this starts a signaling cascade. C-AMP activates protein kinase A. Protein kinase A is going to turn off glycogen synthase and turn on phosphorylase kinase. That's going to activate glycogen phosphorylase, which turns glycogen into glucose. That one epinephrine signal led to 10,000 units of blood glucose. And that's kind of a big deal if you're trying to protect yourself. Now, is this something that we want running all the time? You might be like, oh, it's nice to have glucose out there all the time, nice to have energy all the time. Well, sometimes you want to store some of that energy. So this is going to need to get turned off. Well, how do we turn this thing off? Well, we break down some of those enzymes that we're using. If you make more adenylyl cyclase or break down something called phosphodiesterase, that's going to increase your C-AMP in the cell. If you turn off the G protein or turn on the G protein, that's going to change what happens. Because remember, step A in a cell signal can't happen before or after step B. You got to go A, then B, then C, then D. If you take out one of those steps, that's going to stop the signal completely. And signal regulation happens quite often in our cells because our cells like to be efficient. We don't just want energy all over the place, we want to be able to manage it. So that was kind of a lot. I know that's 15 minutes of stuff right there. Let's look at the 3 things that are going to happen with signals that we talked about with our cells. The first thing, ion channels are going to be open. We talked about ion channels. That's one of the types of cell receptors. You open it up, ions can flow in and out of the membrane because ions can't get past that lipid bilayer, so they need a channel to open up. You can also alter gene expression. That happened with cytoplasmic receptors. Cytoplasmic receptors have a lot to do with turning genes on and off, and as we get later in the year, we'll talk about turning genes on and turning genes off in the gene regulation section. The last thing we can do is alter enzymatic activity. Phosphorylase kinase is a big thing that can alter how our enzymes are working. And if we turn that on or off, that's going to determine how much glycogen and free glucose we have in our blood. Hopefully this makes a little bit more sense. We're going to review some of this stuff in class, and have a good evening.